Welcome back. Today we are diving into a topic that's not just reshaping the world of vaccines, it is totally flipping it on its head. We are exploring the cutting-edge world of inverse vaccination and how it's changing our approach to treating autoimmune diseases. We'll also talk to CancerVax, who are using the immune system in a radically different way, trying to make it attack cancer cells the same way it would fight off a virus such as measles.
And this topic, Connor, this was your request. When we started planning, you were like, let's look into this. And it has been so fun to meet the scientists working on this topic. On one side, you've got inverse vaccines calming down an overactive immune response to prevent it from attacking the body. On the other hand, you have cancer therapies trying to hype up the immune response to eliminate cancer cells.
That's exactly right. And the more and more we understand about the immune system, the more I start to think that it is the most extraordinary evolutionary outcome that we can really have been benefiting from over the thousands of years that we have been human beings, millions of years. Vaccines, as you have never met them before, that is what matters on today's episode of Discovery Matters, brought to you by the fun people at Cytiva.
I'm Larry Steinman. I'm a professor of neurology and pediatrics at Stanford, and for about a decade I was the chair of the immunology department. So I'm a neurologist and immunologist.
Larry endured a personal encounter with polio, and the extraordinary impact of the Salk vaccine has left an indelible mark on his life and his career path.
The best example of a conventional vaccine is the vaccine that influenced me and the scientist who influenced me to go into the fields of neurology and immunology. And that was the Jonas Salk polio vaccine, which became available around 1953 or 1954. I was born in 1947. I went on a vacation when I was a 3-year-old with my grandparents. When I came home to my house in August of 1951, I asked, where is my older sister? And I was told she was in a hospital. So she was on the polio ward of Los Angeles County General Hospital. At a very young age, I came face to face with my sister's illness, and she was on the polio ward, which was a very frightening place. There were children in iron lungs and children walking around with these monstrous braces on their legs. That's a successful example of a conventional vaccine. The polio virus was inactivated so that it would not cause polio, and it essentially wiped polio off the face of the map in places where polio vaccine is available. The first research job I had was at the Salk Institute. I worked with a group of people on what was called the genetic control of the immune response to influenza, which was paving the way for the modern influenza vaccine.
The fact that his first research job was at the Salk Institute, named after the very scientist whose work had such a profound effect on his family, is a testament to the power of scientific discovery.
I know, and Larry's story, it just captures the essence of what a conventional vaccine can achieve, taking a once feared virus such as polio and nearly eradicating it through collective worldwide immunization. I mean, it's one of the great stories in human health.
No doubt, no doubt. These traditional vaccines are essentially helping educate, train, and trigger the immune system to recognize, attack, and kill the pathogen. Seems fairly intuitive, whereas inverse vaccination seems counterintuitive.
And fortunately for all of us, that counterintuitiveness is going to be addressed by Larry because he is an expert on inverse vaccines. They are designed to stop the immune system from attacking the body's own cells and help to treat autoimmune diseases. So, unlike traditional vaccines, such as the polio vaccine, which stimulates an immune response, inverse vaccines work by retraining the immune system to tolerate certain specific antigens rather than attacking them. And they do this by delivering disease-related molecules in a way that signals the body to ignore them. It says,
"Move along. These aren't the droids you're looking for."
Reducing autoimmune reactions without suppressing overall immunity. And this approach has been shown real promise in conditions such as multiple sclerosis and type 1 diabetes.
Okay, it's quite a mind bender. So instead of boosting immunity, you're trying to reduce immunity because the immune system was getting a little bit out of control, getting a little
That's exactly.
happy about...
Yeah, your immune system just unleashes madness on your body, essentially.
The immune system is our Department of Defense. As a neurologist, I'm interested not only in diseases that are caused by a virus like polio, but in diseases that result in an attack by the human immune system against one's own tissue, often the brain tissue. So current therapies for multiple sclerosis is to use very effective drugs that suppress the immune system quite widely. And as we learn more and more about these diseases, we come to know that there are very specific immune attacks against very specific molecules in the nervous system. So the thought was, what if we could just throw the off switch for those unwanted immune responses and leave the immune system available to attack things that we need to attack, like polio and other viruses that might come our way. So this would be an inverse vaccine. Let's make an inverse vaccine, but make it very limited to just shutting down unwanted immune responses and leave the rest of the immune system ready to do its job and fight viruses that can harm us.
It makes it clear why balance is so crucial. If it's too aggressive, it attacks your own body. And if it's too weak, we're vulnerable to infections. I mean, it makes perfect sense. The idea of an inverse vaccine is fascinating, a way to fine-tune the defense system that we have rather than to suppress it entirely.
I mean, it's clever the way Larry calls it the Department of Defense, right? And defense is also about diplomacy.
Exactly.
So that's a really interesting balance.
You can really see the analogies between defense and police forces. You need them highly trained, highly intelligent, making smart decisions.
Right.
What are we going after? What are we not going after?
Right. So what do we know about what triggers the unwanted responses by our immune systems?
One of the revelations when we came into the era of genomics is that there's only a limited number of proteins that we make as humans that a virus will also be making. In other words, we humans have genes, about 20,000 genes encoding proteins, and we can make a limited number of proteins. We use the same structures repeatedly in humans that have been used by viruses, bacteria, lower forms of life, so that often a virus will have in its repertoire of proteins a structure that, at least for a certain stretch of the structure, resembles closely or exactly our own structure.
It's kind of mistaken identity. I mean, another term that's used is molecular mimicry. If we go back to multiple sclerosis, which is the immune system attacking the myelin sheath around the nerve cells, this is what causes multiple sclerosis. Larry's approach was essentially to target the myelin protein and reduce the immune system's response to other myelin components and kind of spread the tolerance.
In the 1900s, we tried to build a myelin mimic that had a few variations. Individuals in the clinical trial started getting immunity to their own myelin proteins. 10 years later, we made what we called an inverse vaccine by encoding the antigen in its DNA sequence, injecting the DNA into a person's arm, and then watching to see whether there was an attenuation of immunity to myelin. And there we did a little better. We saw a reduction in the MRI activity and a reduction in the antibodies that were being made to myelin, but the effect was very mild. Could we do this more effectively and still get the precision we would want in an inverse vaccine? So the precision means we only want to attenuate immunity to unwanted immune responses. The trick is getting it strong enough without making it too strong. But what we did not know back in 1999, going into 2000, was what exactly is being attacked in multiple sclerosis. Recent research that was published first in 2022 showed that we were missing something very important, that everybody who has MS has been infected with a virus called the Epstein-Barr virus, EBV. Research for the 8 years previously showed that there was a molecular mimic between a piece of the Epstein-Barr virus and a piece of a molecule that forms our myelin sheath called glial cam.
It sounds like these evolving approaches have been crucial steps in understanding and treating autoimmune conditions like MS.
Understandably, Larry's paper, published in 2022, made a big splash.
It's important that the work can be validated by some other group. We see, unfortunately, too many times in science where investigators make a big pronouncement and publish a peer-reviewed paper, and then other people try to repeat the work, and they say, well, it only works on a Tuesday. It didn't work on the other 6 days of the week when we tried the experiment. You want something that's robust and reproducible in the hands of others. So this interview is timely because a paper is about to be published. I'm giving this interview in early February, but it will be out in a few weeks. It's been accepted for publication, and a group in Sweden at the Karolinska repeated our results, and they're even more elegant because they used bigger numbers of patients in this study. In Sweden, they have a registry of all the patients in the country who have multiple sclerosis. If you start with having a certain genetic background, and if you make an immune response to Epstein-Barr virus, and if you make an immune response to gliacam, the odds that you're going to get multiple sclerosis compared to a sample of normal people goes up about tenfold.
With the established link between Epstein-Barr virus and multiple sclerosis, along with its association with other conditions, how much is our biotech industry focusing on eradicating EBV?
One of the big projects throughout the pharmaceutical industry that's making both RNA vaccines and more conventional vaccines. So this includes companies like Moderna, Pfizer, Sanofi. All of these companies are hard at work on making an Epstein-Barr virus vaccine. And the hope is that if successful, people won't get mononucleosis, they won't get cancers caused by the Epstein-Barr virus, and multiple sclerosis might someday be something we talk about in the Museum of Diseases that used to affect humanity. And the worry, because of this molecular mimicry, is we better be careful about what we choose in the composition of the vaccine so that we don't get something unwanted, like an outbreak of multiple sclerosis if we choose the wrong components.
Aha! A museum of diseases. I love this. What a great idea. A lovely, lovely idea. Brings us neatly back to the fact that polio has almost been completely eradicated in most Western countries. Do you remember that every day's a school day fact that we had, where we thought suddenly viruses might be one of the causes of Alzheimer's?
Right.
Viruses have been identified as the causes of certain cancers. I mean, this is amazing. We need to do this. I certainly remember polio as a child, having grown up in the global south. I mean, it was everywhere, and I had friends, and there were plenty of adults around who had lived with polio.
Well, you're younger than me.
Just a smidge.
I don't remember polio, but we're not here to talk about the gap in access to therapies in this episode.
Okay.
We have spoken about that before. So let's get back to Larry, and I wonder what surprised him about how the immune system responds to environmental factors and pathogens, and how these effects can resurface years later.
As we understand it now, there's a lot of components of nearly every virus that do have some structural similarity with our own proteins. We have a number of different genes that we're born with that decide what part of a molecule that our immune system is going to attack. So you can make a perfectly effective response against a virus like EBV if you don't happen to have one of those genes that homes in on this region of similarity. And so those genes have been known for a long time. They're called human leukocyte antigens, and they are one of the reasons why we can't transplant organs from people on this podcast willy-nilly. We have to find a good match. So those HLA molecules help decide for the immune system what part of a target to attack. So it gets splendidly complex, but for our own good. By being aware of exactly what part of a self-antigen mimics a virus, we can then attempt to make these precision inverse vaccines just to shut down that one unwanted response. And you could leave the immune system able to attack the rest of the virus because we'd like to be immune to that virus. We just don't want to be immune to that part of the virus shared with our cells or our self-antigens.
How do we introduce innovative approaches in clinical research and medical practices? The medical practices are conservative. Clinical research is conservative for good reasons. Patient safety comes first. So how does Larry cope with failing fast?
I think the general public could not possibly understand how frustrating the work is. Most of the time, experiments don't work. Or you can come to work on a Monday, Tuesday morning with a great idea, and it takes a year or so to prove or disprove the idea. And most of the time, the idea is disproven. So if we want to have a strong system, we should do as much as we can to encourage people to try this kind of career. I mentioned that when I was 17, I got to work with the Salk Institute. I got to meet Jonas Salk. I worked with some equally brilliant scientists, but we need to give these kinds of opportunities and open these doors. But it's a very tough career, so it needs to be encouraged, and we really need to provide resources to enable science, whether it succeeds or fails. You need a few failures. If we're interested in sports, our team doesn't win all the time, and we need to be able to understand why we lost and what could be done to mitigate that.
Navigating these setbacks and uncertainties of scientific research takes real resilience, and it's something that, scientists have to build over their careers. You're going to get knocked back again and again and again. Failure is part of progress in science.
What are the key factors needed to successfully develop inverse vaccines, given the challenges of identifying the right antigens and the risks of failures?
On the subject of inverse vaccines, the more we know about what we should try to eliminate, the better. I told you a couple of attempts. We didn't have the right antigen. We didn't know what the right antigen was, so we chose the most likely suspects. And then there's a number of dazzling technologies available these days, so people have to be bold and do well-designed experiments in humans to show that the ideas can be translated. And hopefully they'll get it right the first time or early on, and there won't be problematic failures.
Where inverse vaccines are designed to suppress specific immune responses, the opposite end of the immunotherapy spectrum tricks the immune system into recognizing cancer cells as foreign entities, similar to pathogens such as measles, and activating an immune response against them. We have an example from CancerVax. Our guides for this are Adam Grant, not that one, and George Katiba.
But speaking of Adam Grant, I thought, it pays to know who you're speaking to. So that was my first question to him is, how often are you confused with the Adam Grant who's famous for insights on workplace and work behavior?
More frequently than you would think. I actually get a lot of credit for some stuff that he said. I make sure to correct everyone, but the only thing I really have on that Adam Grant is I do have a little bit more hair than him. It's nice. If I was to be associated with any other person in the world, I'm glad that's who it is. He's a really great guy.
And then we have George.
I'm the Chief Scientific Officer for CancerVax, and I've worked on cancer and other immunotherapies for actually a couple of decades now. Really passionate about bringing novel and new treatments to patients with serious medical needs there. So I think that's what gets me up in the morning, and love bringing that passion every day to work.
CancerVax is developing a universal cancer immunotherapy platform, which aims to use the body's immune system to target and eliminate cancer cells.
Essentially using the body to cure itself. But frankly, with so many different kinds of cancer, can a universal treatment platform really be achieved for cancer?
It would be amazing, wouldn't it? Like a universal flu vaccine.
Right.
We're buoyed by the success of checkpoint immunotherapy, which really does that. And so it has made really transformative for cancer patients worldwide. And that really is activating the body's own immune system to treat cancer. We're taking a bit of a different approach here about using pre-existing immunity to well-immunize diseases and pathogens and redirecting that against the tumor. So all of our bodies already have this pre-existing immunity. Our immune system is really good at defending us from pathogens there. And so we're hoping to redirect that against cancer. So we do believe that each one of us has this power inside their bodies. We just need to get the body to turn it back on.
Don't expect this one treatment will fit all for all the different cancer types out there. We call it a platform because there's some certain components of this treatment that we can cater towards different cancer types.
That is fascinating. How does the platform adapt to accommodate the different needs of patients with varying types of cancer?
We're trying to utilize a two-step approach to detect cancer cells. And the way we're going about this is what we refer to as like a marker 1 and a marker 2. And our marker 1 focuses on proteins that are on the outside of the cell. Breast cancer and pancreatic cancer are going to have different proteins on the outside of the cancer cell. And so with our platform, we expect that we could be able to tweak our nanoparticle to target proteins that are more biased towards breast cancer or more biased to pancreatic cancer. However, for us, something that we have been really focusing on is to further detect these cancer cells from other different cancer types and healthy cells by using our marker 2, which looks at genetic signatures inside the cancer cell.
So as we look at the evolving landscape of cancer treatment, there is a clear need to expand the reach of effective therapies. While we have seen incredible breakthroughs in certain cancers such as melanoma and kidney cancer and breast cancer, many patients with other types of cancer such as pancreatic cancer still face very limited options.
We are validating our therapeutic hypotheses in preclinical models, and we are focused currently on pancreatic cancer. We believe there's a really urgent and high unmet medical need there for novel therapies for patients. It's a really devastating disease where treatments really have not advanced meaningfully. There's some bright things on the horizon coming, with maybe some of these KRAS inhibitors really can make a difference here. But we really need these potent treatments like immunotherapies that give long-lasting and durable remissions to patients and improve their quality of life. What we're trying to do is recreate some of that success that you're seeing now, where it turns into a chronic disease, or people are always reticent to use the cure word here. But we're looking decades out on some of these patients from the initial immunotherapies here. And that's what we're hoping to drive in a broader swath of patients. We've seen great success in things like melanoma. It's really transformed the disease there. But, these are still a minority of patients, and we want to really bring the benefits of immunotherapy and the power of the body's immune system to a broader audience, to more of those patients.
Especially patients that have more of a cold tumor microenvironment, where they might not have the readily available immune infiltration in the tumor microenvironment. But we're hoping that with our approach, tricking the immune system into killing cancer cells by making it look like something the body already recognizes, like measles, is that we can not only have those immune cells kill the cancer cells, but also convert the tumor microenvironment from a cold to a hotter tumor microenvironment to elicit that immune response.
I think if I hear it right, what Adam is saying here really captures the core of why immunotherapy can be so powerful, but also why it's not a one-size-fits-all solution. Immunotherapy has revolutionized the treatment of cancers like melanoma, but for a lot of other cancers, the challenge is that these cells are basically experts at hiding. They are biological stealth operators.
Yeah, they are like the Jedi mind trick people of the immune system. The immune system just doesn't see them as threats, so it doesn't respond. And what Adam's team is doing with their platform is essentially handing the immune system a map or an identity card and marking those hidden cancer cells as targets by making them look like something that it already knows how to fight, such as measles. It's just the most fascinating approach.
Wow. And so as new treatments like immunotherapies come to market, the challenge then becomes ensuring that they are available to those who need them most, especially given the costs involved. So I want to hear Adam and George's perspective on how CancerVax plans to address this issue and what they're doing to make life-saving treatments more accessible.
Current treatments that are being explored right now, things like CAR T cells and personalized cancer vaccines, they rely on a very time-consuming process of using the patient's own immune cells. By using those cells from the patient, by using that genetic information, it increases the cost hugely. It's a personalized one-off treatment that's being developed, and they have been effective. It's very laborious to make. You're not going to see this outside of major academic medical centers. It's just not feasible for the majority of patients. Here, we're hoping to utilize really the infrastructure that was developed for a lot of the COVID vaccines here. So we're using tried and true liposome technology there. We need to tailor it for a patient's general cancer type, but not the individual cancer type. And so we hope it to be an off-the-shelf therapy there that we can also manufacture at a lower cost.
It's scary. Even though these personalized approaches have had great success, there's still so much variability in them. You don't know if they're going to work for you or not. There's no guarantee, and that's after you've already paid for it. I think that's why a lot of people are hesitant, and even insurance companies. But with us having that more off-the-shelf approach that is not only more available, but hopefully more efficacious for cancer patients because we're utilizing their natural immune system.
What's next, and what hurdles does CancerVax need to overcome before achieving their mission?
Science is challenging, and this is what gets us up every day and keeps us motivated, is overcome these challenges to really prove our therapy and to get it into the point where we're able to test it and bring it to clinical trials there. Right now, we are a preclinical company, and our next steps will be to prove this in preclinical models, show that we can actually induce durable anti-tumor activity in our preclinical models, and that will hopefully set us up to bring this therapy towards the medical regulators for approval to go into human trials.
We've actually have made a lot of progress at CancerVax so far. We've just recently showed that our smart mRNA technology is able to, distinguish between healthy cells and cancer cells at more of a synthetic level. But that was the first step we need to show. Can we actually use these internal genetic signatures to distinguish between healthy cells and cancer cells to further reduce potential off-target effects? We're developing a targeted nanoparticle, which we're really excited about, and we think is going to be a key aspect of our platform. We got a lot to do, but luckily, we've already been working really hard on this project for quite a while.
So by making cancer cells resemble something the immune system already knows how to fight, you can bypass some of the common challenges that traditional immunotherapies face, such as tumors finding ways to hide from immune detection. And this strategy could open doors for treating cancers that have been particularly resistant to existing immunotherapies.
Other immunotherapies try to modulate the immune system or help stimulate the immune system to attack cancer cells. We're actually really unique in the sense that we are trying to trick the immune system. We've realized that the body is already so good at killing pathogens that it recognizes. And so we're trying to induce this immune response by actually making the cancer cells look like something that the body already recognizes, and no other therapy is taking this approach. And we believe that there's a lot of benefits to this approach, such as it can be more off the shelf. We are hoping that we can elicit a stronger immune response, and also that this therapy could potentially benefit cancer types that don't already respond to immunotherapies, such as pancreatic cancer. And that's one of the main cancer type that we're focusing on right now.
In 4 or 5 years, let's say CancerVax perhaps has successfully introduced its cancer vaccine platform. What does the world look like?
4 or 5 years from now on cancer treatments, it's going to be a whole different landscape. You're going to see a lot more targeted modalities brought there, a lot more combination therapies. Hopefully immunotherapies like our universal cancer treatment are really going to be at the forefront of these medical therapies here. So things that you're going to get as a standard when you're diagnosed with these diseases, they'll pull something off the shelf for you, give you that treatment, and combine it with big data-based approaches, knowing which treatments combine with your tumors, using a universal cancer treatment like ours as the backbone of a platform, and then bolting on to that drugs that match your genetic signature. Personalized medicine may already be here, where it's matching your tumor that you get diagnosed with, matching you with a drug like Herceptin. That is personalized medicine at a large scale. It's just how do we expand that broadly and go from a single test to matching one drug to understanding your genomic signature holistically and then matching that to a combination set of therapies. I think we're very close, actually.
I really think that these mRNA-based therapies are really going to change the game. And that's what ours is, utilizing our smart mRNA to make the cancer cells look like something like measles, tricking the immune system. And the thing is, it's just so great about mRNAs. It's just much more natural than any other therapy. It's not like a harmful chemotherapy drug. Everyone has trillions of mRNA in them at any time, gazillions, maybe. What we've learned about biology and the different capabilities we have with next generation sequencing and using these data sets is that we can really cater these much more natural, less harmful, much more hopefully efficacious George and I waking up every day, and we love working at CancerVax. It's such a great opportunity to be in this forefront of this shift of this technology, and just the potential is just unbelievable.
While the CancerVax approach and inverse vaccines are on opposite ends of the immunotherapy spectrum, they both use the body's immune system to achieve their goals. The CancerVax platform attempts to enhance immune recognition of cancer cells, whilst inverse vaccines seek to reduce or regulate immune activity to prevent harmful self-directed attacks.
In the broader context of vaccines, this conversation touches on evolving understanding of immunotherapy and how advancements in mRNA technology, big data, and genetic analysis are expanding the possibilities of personalized medicine at scale. Very important. Both cancer immunotherapies and inverse vaccines represent cutting-edge strategies for reprogramming the immune system to achieve desired outcomes. Super fascinating stuff. Like you said, Connor, whether to destroy harmful cells or protect the healthy ones from misguided attacks.
Nobody likes friendly fire.
Right.
Right. Okay.
Get in touch with us at discovery.matters@cytiva.com. Our producer is Beth Armit-Brewster. Editing, mixing, and supervision by Tom Henley. My name is Dodie Axelson.
And I'm Connor McKechnie. Make sure you rate us on Spotify or whichever platform you use. We'll see you when we come back with another episode of Discovery Matters.